Record. Right. When we study this thylakoid structure, it is a membrane-bound small disc. That means this structure is a little bit unique chloroplast because inside again we can find membrane-bound sub-organelles. Most of the organelles, they don't have this kind of sub-organelles. If you take Golgi, endoplasmic reticulum, ribosome, nucleus, mitochondria, we don't get this kind of structures. But if we look what we have learned from the beginning now, mitochondria and the chloroplast were prokaryotes, and they incorporated into the large cells to become eukaryotic cells. They also change their structures to become evolved. Prokaryotes has cell wall, but mitochondria and chloroplast, they don't have cell wall. But they change according to the cells. As a result, the thylakoid become one of the most important structure in the chloroplast. And large number of thylakoids we can find. They can provide more surface area by making these membrane-bound small structures called thylakoid. Because membrane can increase the surface area. Because I can find large amount of cell membrane, this can increase the surface area inside the chloroplast. Okay, did you copy this one?
Yes, sir.
Right. Then we trying to see why thylakoids are important. Okay. Let's see like this. Thylakoid is a disc like this, and this there are many thylakoids make these columns called granule. And if you take one thylakoid, so we take a cell membrane of this one, and as a typical cell membrane, it is phospholipid bilayer. Right, when we are learning cell membrane, we learn this has proteins. Cell membrane chiefly it's a phospholipid bilayer, and the proteins are embedded to make many different functions, because some proteins are enzymes, some proteins are transporters, some proteins are receptors. So we learn all the function of these proteins. Now here similarly we can also find a protein which is again embedded inside this phospholipid bilayer. Right now we are learning on the notes section, page 4, photosystem. Okay, this is the part of the cell membrane of this thylakoid. But you can see phospholipid bilayer, and there are two protein complexes. Now, why these protein complexes are important? These protein complexes are made with collection of different proteins, molecules, and also pigments. Let's see, the red color one represent the protein molecules like this, and then white color one shows the pigments, and also the green color one, and maybe the yellow color one. Right now we can see a collection of proteins and collection of pigments. So we write it here. The red color ones are proteins. Rest of the things, green color and white color ones are pigments. So protein plus pigments, they make complete this protein structure, and this aggregation of protein is called the antenna complex. That is an aggregation of proteins. Now, aggregation of proteins and pigments make this photosystem. So we abbreviated with PS, photosystems. Now, pigments are two types. We can find the primary pigments, and the other one is called accessory pigments. So A, primary pigments, B, accessory pigments. Now, why we have two different pigments? Because it is a color and it is a role in the photosynthesis. We divide these pigments into two. So primary pigments are chlorophylls. Plant has again two types of chlorophylls, chlorophylls A and chlorophylls B. Chlorophyll A and chlorophyll B. And chlorophyll A and chlorophyll B are the main pigments which provides a significant contribution for the photosynthesis. And accessory pigments, they have a different colors. They could be yellow color, they could be a gray color. They are carotenoid and phycobility proteins. So carotenoids are usually the orange color, yellow color pigments. Right. They're also important for the photosynthesis. They can also contribute for the photosynthesis. But there is a difference between primary and accessory pigments. Primary pigments dominating, and their contribution is very significant for photosynthesis. We'll see later on why they are important. But the major role of this primary and accessory pigment, they can absorb energy from the sunlight. The primary pigments and accessory pigments has the ability to absorb energy from sunlight. Right now, if I show this here, these arrows represent the sunlight, and after that, proteins and all the pigments, they help to absorb the sunlight, and then the energy of the sunlight finally focusing to the bottom of this photosystem, and that is called a reaction center. Reaction center. Right, on the note section, you can find this information, and we have to add this diagram, and we can see all things are discussed. What is missing still from this picture? There are two type of photosystem. Now you see here the two photosystems. The first photosystem is labeled as a photosystem 1. The second one is also labeled as photosystem 2. And there's another name given that is called photosystem 1 is called 700 nanometer photosystem, and this one is called 680 nanometer. So why they have a different wavelength given to label the photosystem? Then the answer is coming to the next topic, that is sunlight energy, light spectrum. So we have to look a little bit about the light spectrum and why the light spectrum has different range, and these photosystem collectively, the primary, accessory and proteins can absorb largely light ranging from 700 nanometer. That means they are specialized in the range of 700 nanometer. And this one specialized for 600 nanometer, the wavelength of 600 nanometer. That's why PS1 and PS2 photosystem important. Okay, just to summarize, thylakoid is the small membrane bound disc inside the chloroplast, and the cell membrane made out of phospholipids. And the phospholipid membranes, we can find photosystem complexes. Photosystem complexes are two types, photosystem 1 and photosystem 2. Photosystem consists of proteins. Aggregation of proteins are known as antenna complex, and collectively protein and pigments make photosystems. And pigments are two types, primary pigments, chlorophyll A and chlorophyll B. Accessory pigments, there are many names can be given. Example, carotenoids. There are carotenoids and xanthophyll, phycobilioproteins, like many pigments, different color pigments can be present in plant cells. Okay, next we're going to learn about this spectrum, light spectrum, and why this light spectrum is important, and why we have two different photosystems. Did you copy this one?
Yes.
Right, then we are going to the electromagnetic spectrum. So the sunlight energy reaching from sun onto the earth as radiations, because the heat energy is generated from this nuclear reaction, that is a fusion of hydrogen atoms, hydrogen nucleus fused to make the helium. This release large amount of energy as heat energy. But if you look sun and earth, there is a vacuum between the sun and earth on the space. So then conduction, convection and radiation, if we take these three methods, conduction and convection, they need either solid surface or air. So that two methods cannot use to transfer the heat energy. Only the radiation is the mode of transmission of this heat energy. And we consider these electromagnetic radiations. There are different wavelengths we can find, right? So we have a gamma rays, X-rays, UV, IR, microwave, FM, AM, long radio waves. It depends on the length of the wavelength, right? So, for example, these they have very short wavelengths, and they are very powerful in their energy. Especially gamma rays are very powerful because they have very short wavelength, and the X-rays, then again UV, and then this is a visible light, and after that we can find IR rays. Then it's a wavelength is increasing like this. Now, when we consider sunlight energy, mostly uh, UV radiation are filtered by the ozone layer, and there are two UV radiations, UVA and B, and certain amount of UV radiation escape from this layer, and it is reaching to the earth. But uh, the most of the sunlight coming as a visible spectrum. After that, uh, this visible spectrum brings heat energy and also light energy. So once it touched the surface on the earth, again it's reflecting as infrared radiations back to the atmosphere. So like this, if the earth's surface, so when this UV light hit the surface, and some of the light is passing as infra IR radiations. So then we can find the greenhouse gases, and again some of them reflecting back. So collectively, this one and this one good enough to provide the right temperature on Earth. So what we talking about as a global warming, if this layer getting thicker due to accumulation of methane and carbon dioxide, more light is reflecting back. So when more light is reflecting, it can increase the temperature on the surface. That's called global warming, right? It's not important for here. We can learn under that uh, the environment, ecosystems in next other chapters. But the visible light is the light which usually we get, and it has a heat and a light energy. Now, visible light has 400 to 700 nanometer range. And what are the colors? Blue, actually it's violet, indigo blue, then more towards the green, then yellow, orange and red. And this area is more blue, and this area is more red, and this middle is more green. And it consists of these 7 colors, but 400 to close to 500 is more blue color, 500 600 is a green color, 600 700 is a red color. Right now, how these photosystems are important in absorbing the light. Within 400 to 700, these pigments, different pigments, accessory pigment, primary pigments, together with the proteins, they are capable of absorbing the light. Okay, let's see what is the exact amount of light can be absorbed by these pigments. Right, this one is an important note to copy down. So I'm going to draw a 2 graph like this. Okay, now y-axis, first graph, I'm going to label as a percentage of absorption, right? It's percentage of light absorbed, amount of light absorbed, percentage of light absorbed. And the x-axis, 400 nanometer, 700 nanometer, the wavelength of the visible light, right? X-axis is the wavelength of the visible light. I've seen just right now. If I take chlorophyll A, now chlorophyll A can absorb light, highest this, and it's getting low, and again here it can absorb light. So if we look at this point, let's say 1 point, the given wavelength, this is the amount of percentage of light absorbed by the chlorophyll A. Let's take a chlorophyll B. So chlorophyll B can also absorb in this range, and again it's decreasing here, and then again it can absorb here. So this is B, this one is A. Right, if you take a carotenoid as one example as accessory pigment, that can also absorb light, at this, and then again like this. Okay, if we see this area is a blue color area and this area is a red color area and this area is a green color area. Now largely light is absorbed blue color area and the red color area. Now green color area, very less amount of light is absorbed. That means light is reflect or emit. Green color range is not absorbed, instead it emit or reflect. This is the reason we can see plant as a green color. Why plants are green color? Because they reflect green color light. They don't absorb it. So blue and red color is mainly absorbed. So can we write here now? This graph is showing the amount of light absorbed at each wavelength. Amount of light absorbed at each wavelength. So this graph is named as absorption spectrum. The name given to this graph is absorption spectrum. And definition of absorption spectrum is amount of light absorbed at each wavelength. Amount of light absorbed at each wavelength. Alright, now we have a second graph. Second graph again, we can place a wavelength on x-axis. Now on the y-axis, we're going to add different label, that is a rate of photosynthesis. Right, now if we want to add the rate of photosynthesis by looking at the first graph, we can make a conclusion. If the amount of light absorbed is higher, then the rate of photosynthesis is high. Rate of photosynthesis is higher because more energy absorbed, the rate is higher. So that means this level rate is the highest, and then the second highest coming for this. That means the graph is very close to this graph, so it's a single line, so we can show it like this. So we can write it rate of photosynthesis at each wavelength. and this is called action spectrum. Absorption spectrum and the action spectrum. Absorption spectrum is amount of light absorbed at each wavelength. Action spectrum is a rate of photosynthesis at each wavelength. And they look a little bit similar. The reason is if the higher light absorbed, rate is higher. Right, you can copy this down to the page under the absorption spectrum and action spectrum. And it's also given the definitions on the notes section, absorption spectrum and action spectrum. Did you copy this?
Yes.
Right, we have done the basic information required to understand the main concept of photosynthesis. So what is the basic information so far we learned? The chloroplast structure and the thylakoid, importance of thylakoid in terms of photosystems and the structure of photosystems. And after that, last thing we learned, the photosystem has pigments that can absorb light, and the light absorption is depend on the type of pigments. So they can absorb mostly blue color and red color of the light. So when different composition of pigments there, they can absorb different light. As a result of that, we have a PS 680 and PS 700, two type of photosystems. Their energy absorption level is different. As a result, if they can absorb different amount of energy, their function is also different, because one get higher energy, one get a little bit lower energy. Based on this energy level, they have different role in the photosynthesis. Right, next lesson on the notes, it says light dependent reaction, non-cyclic photophosphorylation. Right, let's learn what is this light dependent reaction, non-cyclic photophosphorylation. Light dependent reaction. This indicate the reaction need light. It happens if light is present, that means it happened during the daytime. And the next couple to this topic is non-cyclic photophosphorylation. Now, what is non-cyclic? It is not happening in a cyclic way, because we learned Krebs cycle. The Krebs cycle is it's always substrate adding to the one place, and then it goes as a cycle. But when it's a non-cyclic, just like a glycolysis, we learned glycolysis. Glucose is the starting substrate, and the end product is pyruvate. It is going only one direction, and it doesn't have a cyclic pattern. And what is photophosphorylation? We learned previously oxidative phosphorylation. So oxidative phosphorylation, what we learn, ADP molecule can combine with a phosphate group to make ATP. And here, in electron transport chain and oxidative phosphorylation, we have seen energy of the glucose molecules used to combine ADP molecule with the phosphate group. That means glucose energy is provided at this point to make ATP molecule, and that is called oxidative phosphorylation. What is the meaning of photophosphorylation? Phosphorylation is adding a phosphate group to ADP molecule to make ATP molecule. Then the difference is actually this word photo. So we can understand now the energy is provided by the light. So light energy is provided to combine ADP molecule with phosphate group to make ADP molecule. So then if we look at this topic, light dependent reaction, we can understand it is something to do with making ATP molecule. That means light energy is used to make ATP molecule. So we are going to learn this reaction in this topic, light dependent reaction, non-cyclic photophosphorylation. So photophosphorylation is adding a phosphate group to ADP molecule using the energy of the sunlight. Okay, so I need the space on the screen. So I'm going to delete this title. It's on the note section, light dependent reaction, non-cyclic photophosphorylation. Okay, we try to understand this process. So I am going to draw a cell membrane of the thylakoid like this. So this box-like structure represents the thylakoid membrane. And I'm going to add, you can also do the same time, one by one. Then I'm going to add one photosystem here. Let's say this is a one photosystem, and here another photosystem. So these are the two main photosystems, and we can label them as PS2 and PS1. So we name first PS2, not PS1, and then we label PS1. Why is that? Because starting point of the light dependent reaction start at PS2. So PS2 is a nanometer 680 and this is a nanometer 700. Right, after that there are other protein complexes located inside this membrane, and they are also important for the light dependent reaction. But we don't want to know the names of these proteins. There's a one here, another one here, another one here. And photosystem 1 also has 1, 2 And third one.